Disease models. Animal models serving in research may have an existing, inbred or induced disease or injury that is similar to a human condition. These test conditions are often termed as animal models of disease. The use of animal models allows researchers to investigate disease states in ways which would be inaccessible in a human patient, performing procedures on the non-human animal that imply a level of harm that would not be considered ethical to inflict on a human. The best models of disease are similar in etiology, mechanism of cause, and phenotype, signs and symptoms, to the human equivalent. However, complex human diseases can often be better understood in a simplified system in which individual parts of the disease process are isolated and examined. For instance, behavioral analogs of anxiety or pain in laboratory animals can be used to screen and test new drugs for the treatment of these conditions in humans. A 2000 study found that animal models coincided on true positives and false negatives, with human toxicity in 71% of cases, with 63% for non-rodents alone and 43% for rodents alone. 66. In 1987, Davidson et al. Suggested that selection of an animal model for research be based on 9 considerations. These include 1. Appropriateness as an analog. 2. Transferability of information. 3. Genetic uniformity of organisms, where applicable. 4. Background knowledge of biological properties. 5. Cost and availability. 6. Generalizability of the results. 7. Ease of and adaptability to experimental manipulation. 8. Ecological consequences. And 9. Ethical implications. 67. Animal models can be classified as homologous, isomorphic or predictive. Animal models can also be more broadly classified into 4 categories. 1. Experimental. 2. Spontaneous. 3. Negative. 4. Orphan. 68. Experimental models are most common. These refer to models of disease that resemble human conditions in phenotype or response to treatment but are induced artificially in the laboratory. Some examples include the use of metrazole pentylnetetrazole as an animal. These refer to models of disease that resemble human conditions in phenotype or response to treatment but are induced artificially in the laboratory. Some examples include the use of metrazole pentylnetetrazole as an animal model of epilepsy. 69. Induction of mechanical brain injury as an animal model of post-traumatic epilepsy. 70. Injection of the neurotoxin 6-hydroxydopamine to dopaminergic parts of the basal ganglia as an animal model of Parkinson's disease. 71. Immunization with an autoantigen to induce an immune response to model autoimmune diseases such as experimental autoimmune encephalomyelitis. 72. Occlusion of the middle cerebral artery as an animal model of ischemic stroke. 73. Injection of blood in the basal ganglia of mice as a model for hemorrhagic stroke. 74. 75. Sepsis and septic shock induction by impairing the integrity of barrier tissues, administering live pathogens or toxins. 76. Infecting animals with pathogens to reproduce human infectious diseases. Injecting animals with agonists or antagonists of various neurotransmitters to reproduce human mental disorders. Using ionizing radiation to cause tumors. Using gene transfer to cause tumors. 77. 78. Implanting animals with tumors to test and develop treatments using ionizing radiation genetically selected, such as in diabetic mice also known as NOD mice. 79. Various animal models for screening of drugs for the treatment of glaucoma. The use of the ovariectomized rat in osteoporosis research. Use of Plasmodium yoelli as a model of human malaria. 80, 81, 82. Spontaneous models refer to diseases that are analogous to human conditions that occur naturally in the animal being studied. These models are rare, but informative. Negative models essentially refer to control animals, which are useful for validating an experiment. Spontaneous models refer to diseases that are analogous to human conditions that occur naturally in the animal being studied. These models are rare, but informative. Negative models essentially refer to control animals, which are useful for validating an experimental result. Orphan models refer to diseases for which there is no human analog and occur exclusively in the species studied. 68. The increase in knowledge of the genomes of non-human primates and other mammals that are genetically close to humans is allowing the production of genetically engineered animal tissues, organs and even animal species which express human diseases, providing a more robust model of human diseases in an animal model. Animal models observed in the sciences of psychology and sociology are often termed animal models of behavior. It is difficult to build an animal model that perfectly reproduces the symptoms of depression in patients. Depression, as other mental disorders, consists of endophenotypes 83 that can be reproduced independently and evaluated in animals. An ideal animal model offers an opportunity to understand molecular, genetic and epigenetic factors that may lead to depression. By using animal models, the underlying molecular alterations and the causal relationship between genetic or environmental alterations and depression can be examined, which would afford a better insight into pathology of depression. In addition, animal models of depression are indispensable for identifying novel therapies for depression. 84, 85. Important model organisms. Model organisms are drawn from all 3 domains of life, as well as viruses. The most widely studied prokaryotic model organism is Escherichia coli, E. coli, which has been intensively investigated for over 60 years. It is a common, gram-negative gut bacterium which can be grown and cultured easily and inexpensively in a laboratory setting. It is the most widely used organism in molecular genetics, and is an important species in the fields of biotechnology and microbiology, where it has served as the host organism for the majority of work with recombinant DNA. 86. Simple model eukaryotes include baker's yeast, Saccharomyces cerevisiae, and fission yeast, Schizosaccharomyces pomba, both of which share many characters with higher cells, including those of humans. For instance, many cell division genes that are critical for the development of cancer have been discovered in yeast. Chlamydomonas reinhardtii, a unicellular green alga with well-studied genetics, is used to study photosynthesis and motility. C. reinhardtii has many known and mapped mutants and expressed sequence tags, and there are advanced methods for genetic transformation and selection of genes. 87. Dictyostelium discoideum is used in molecular biology and genetics, and is studied as an example of cell communication, differentiation, and programmed cell death. Laboratory mice, widely used in medical research. Among invertebrates, the fruit fly Drosophila melanogaster is famous as the subject of genetics experiments by Thomas Hunt Morgan and others. They are easily raised in the lab, with rapid generations, high fecundity, few chromosomes, and easily induced observable mutations. 88. The nematode Caenorhabditis elegans is used for understanding the genetic control of development and physiology. It was first proposed as a model for neuronal development by Sidney Brenner in 1963, and has been extensively used in many different contexts since then. 89. 90. C. elegans was the first multicellular organism whose genome was completely sequenced, and as of 2012, the only organism to have its connectome neuronal wiring diagram completed. 91. 92. Arabidopsis thaliana is currently the most popular model plant. Its small stature and short generation time facilitates rapid genetic studies. 93. And many phenotypic and biochemical mutants have been mapped. 93. A thaliana was the first plant to have its genome sequenced. 93. Among vertebrates, guinea pigs Cavia porcellus were used by Robert Koch and other early bacteriologists as a host for bacterial infections, becoming a byword for laboratory animal, but are less commonly used today. The classic model vertebrate is currently the mouse Mus musculus. Many inbred strains exist, as well as lines selected for particular traits, often of medical interest, e.g. body size, obesity, muscularity, and voluntary wheel running behavior. 94. The rat Rattus norvegicus is particularly useful as a toxicology model, and as a neurological model and source of primary cell cultures, owing to the larger size of organs and suborganellar structures relative to the mouse, while eggs and embryos from Xenopus tropicalis and Xenopus laevis, African clawed frog, are used in developmental biology, cell biology, toxicology, and neuroscience. 95. 96. Likewise, the zebrafish Danio rario has a nearly transparent body during early development, which provides unique visual access to the animal's internal anatomy during this time period. Zebrafish are used to study development, toxicology and toxicopathology. 97. Specific gene function and roles of signaling pathways. Other important model organisms and some of their uses include T4 phage, viral infection, tetrahymenodermophila, intracellular processes, maize, transposons, hydras, regeneration and morphogenesis. 98. Cats, neurophysiology, chickens, development, dogs, respiratory and cardiovascular systems, Nothobranchius frazeri, aging. 99. Non-human primates such as the rhesus macaque and chimpanzee, hepatitis, HIV, Parkinson's disease, cognition, and vaccines, and ferrets, SARS-CoV-2. 100. Selected model organisms. The organisms below have become model organisms because they facilitate the study of certain characters or because of their genetic accessibility. For example, E. coli was one of the first organisms for which genetic techniques such as transformation or genetic manipulation has been developed. The genomes of all model species have been sequenced, including their mitochondrial chloroplast genomes. Model organism databases exist to provide researchers with a portal from which to Anolis carolinensis carolina anole reptile biology, evolution Mus musculus house mouse mammal disease model for humans Gallus gallus red jungle fowl bird. Embryological development and organogenesis. Taniopygia castanotis. Australian zebra finch bird vocal learning, neurobiology. 117. Xenopus laevis. Xenopus tropicalis. 118. African clawed frog. Western clawed frog amphibian embryonic development. Many animal models serving as test subjects in biomedical research, such as rats and mice, may be selectively sedentary, obese and glucose intolerant. This may confound their use to model human metabolic processes and diseases as these can be affected by dietary energy intake and exercise. 119. Similarly, there are differences between the immune systems of model organisms and humans that lead to significantly altered responses to stimuli. 120, 121, 122, although the underlying principles of genome function may be the same. 122. The impoverished environments inside standard laboratory cages deny research animals of the mental and physical challenges are necessary for healthy emotional development. 123. Without day-to-day variety, risks and rewards, and complex environments, some have argued that animal models are irrelevant models of human experience. 124. Mice differ from humans in several immune properties. Mice are more resistant to some toxins than humans, have a lower total neutrophil fraction in the blood, a lower neutrophil enzymatic capacity, lower activity of the complement system, and a different set of pentraxins involved in the inflammatory process, and lack genes for important components of the immune system, such as IL-8, IL-37, TLR10, ICAM3, etc. 76. Laboratory mice reared in specific pathogen-free (SPF) conditions usually have a rather immature immune system with a deficit of memory T cells. These mice may have limited diversity of the microbiota, which directly affects the immune system and the development of pathological conditions. Moreover, persistent virus infections, for example, herpes viruses, are activated in humans, but not in SPF mice, with septic complications and may change the resistance to bacterial co-infections. Dirty mice are possibly better suitable for mimicking human pathologies. In addition, inbred mouse strains are used in the overwhelming majority of studies, while the human population is heterogeneous, pointing to the importance of studies in interstrain hybrid, outbred, and nonlinear mice. 76. Limitations. Unintended bias. Some studies suggest that inadequate published data in animal testing may result in irreproducible research, with missing details about how experiments are done omitted from published papers or differences in testing that may introduce bias. Examples of hidden bias include a 2014 study from McGill University in Montreal, Canada, which suggests that mice handled by men rather than women showed higher stress levels. 125, 126, 127. Another study in 2016 suggested that gut microbiomes in mice may have an impact upon scientific research. 128. Alternatives. Ethical concerns, as well as the cost, maintenance and relative Ethical concerns, as well as the cost, maintenance and relative inefficiency of animal research has encouraged development of alternative methods for the study of disease. Cell culture, or in vitro studies, provide an alternative that preserves the physiology of the living cell, but does not require the sacrifice of an animal for mechanistic studies. Human, inducible pluripotent stem cells can also elucidate new mechanisms for understanding cancer and cell regeneration. Imaging studies, such as MRI or PET scans, enable non-invasive study of human subjects. Recent advances in genetics and genomics can identify disease-associated genes, which can be targeted for therapies. Many biomedical researchers argue that there is no substitute for a living organism when studying complex interactions in disease pathology or treatments. 129, 130. Ethics. Debate about the ethical use of animals in research dates at least as far back as 1822, when the British Parliament, under pressure from British and Indian intellectuals, enacted the first law for animal protection preventing cruelty to cattle. 131. This was followed by the Cruelty to Animals Act of 1835 and 1849, which criminalized ill-treating, over-driving, and torturing animals. In 1876, under pressure from the National Anti-Vivisection Society, the Cruelty to Animals Act was amended to include regulations governing the use of animals in research. This new act stipulated that 1. Experiments must be proven absolutely necessary for instruction, or to save or prolong human life. 2. Animals must be properly anesthetized. And 3. Animals must be killed as soon as the experiment is over. Today, these 3 principles are central to the laws and guidelines governing the use of animals in research. In the U.S., the Animal Welfare Act of 1970, see also Laboratory Animal Welfare Act, sets standards for animal use and care in research. This law is enforced by APHIS's Animal Care Program. 132. In academic settings in which NIH funding is used for animal research, institutions are governed by the NIH Office of Laboratory Animal Welfare (OLA). At each site, OLA guidelines and standards are upheld by a local review board called the Institutional Animal Care and Use Committee (IACUC). All laboratory experiments involving living animals are reviewed and approved by this committee. In addition to proving the potential for benefit to human health, minimization of pain and distress, and timely and humane euthanasia, experimenters must justify their protocols based on the principles of replacement, reduction and refinement. 133. Replacement refers to efforts to engage alternatives to animal use. This includes the use of computer models, non-living tissues and cells, and replacement of higher order animals, primates and mammals with lower order animals, e.g. cold-blooded animals, invertebrates wherever possible. 134. Reduction refers to efforts to minimize number of animals used during the course of an experiment, as well as prevention of unnecessary replication of previous experiments. To satisfy this requirement, mathematical calculations of statistical power are employed to determine the minimum number of animals that can be used to get a statistically significant experimental result. Refinement refers to efforts to make experimental design as painless and efficient as possible in order to minimize the suffering of each animal subject. See also: animals in space, animal testing on invertebrates. Drosophila melanogaster. Drosophila melanogaster is a species of fly, an insect of the order Diptera, in the family Drosophilidae. The species is often referred to as the fruit fly or lesser fruit fly, or less commonly the vinegar fly, pumice fly, a 5 or banana fly. 6. In the wild, D. melanogaster are attracted to rotting fruit and fermenting beverages, and are often found in orchards, kitchens and pubs. Starting with Charles W. Woodworth's 1901 proposal of the use of this species as a model organism, 7-8 D. melanogaster continues to be widely used for biological research in genetics, physiology, microbial pathogenesis, and life history evolution. D. melanogaster was the first animal to be launched into space in 1947. 9. As of 2017, 6 Nobel Prizes have been awarded to drosophilists for their work using the insect. 10. 11. Drosophila melanogaster is typically used in research owing to its rapid life cycle, relatively simple genetics with only 4 pairs of chromosomes, and large number of offspring per generation. 12. It was originally an African species, with all non-African lineages having a common origin. 13. Its geographic range includes all continents, including islands. 14. D. melanogaster is a common pest in homes, restaurants, and other places where food is served. 15. Flies belonging to the family Tephrididae are also called fruit flies. This can cause confusion, especially in the Mediterranean, Australia, and South Africa, where the Mediterranean fruit fly Cerathitis capitata is an economic pest. Etymology. The term, Drosophila, meaning, dew-loving, is a modern scientific Latin adaptation from Greek words drosos, dew, and philia, lover. The term, melanogaster, meaning, blackbelly, comes from ancient Greek melos, black, and gaster, belly. Physical appearance. Unlike humans, the sex and physical appearance of fruit flies is not influenced by hormones. 16. The appearance and sex of fruit flies is determined only by genetic information. 16. Female fruit flies are substantially larger than male fruit flies, with females having bodies that are up to 30% larger than an adult male. 17. 18. Wild-type fruit flies are yellow-brown, with brick-red eyes and transverse black rings across the abdomen. The black portions of the abdomen are the inspiration for the species name, melanogaster equals, black-bellied. The brick-red color of the eyes of the wild-type fly are due to two pigments. 19. Xanthometin, which is brown and is derived from tryptophan, and drosopterins, which are red and are derived from guanosine triphosphate. 19. They exhibit sexual dimorphism. Females are about 2.5 mm, 0.10 in long, males are slightly smaller. Furthermore, males have a cluster of spiky hairs, claspers, surrounding the reproducing parts used to attach to the female during mating. Extensive images are found at flybase. 20. Female left and male right, D. melanogaster. Drosophila melanogaster can be distinguished from related species by the following combination of features. Gena tilde 1/10 diameter of eye at greatest vertical height, wing hyaline and with coastal index 2.4, male protarsus with a single row of approximately 12 setae forming a sex comb, male epandrial posterior lobe small and nearly triangular, female abdominal tergite 6 with dark band running to its ventral margin, female oviscap small, pale, without dorsodistal depression and with 12 to 13 peg-like outer obisoncilla. 21, 22. Drosophila melanogaster flies can sense air currents with the hairs on their backs. Their eyes are sensitive to slight differences in light intensity and will instinctively fly away when a shadow or other movement is detected. 23. Drosophila melanogaster. Fruit fly feeding off a banana. Scientific classification domain: Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta order, Diptera family, Drosophilidae genus, Drosophila subgenus, Sophophora species group, Melanogaster species. Subgroup: Melanogaster. Species complex: Melanogaster. Species: D. melanogaster binomial name. Drosophila melanogaster. Migan, 1831. Egg of D. melanogaster. Under optimal growth conditions at 25 degrees Celsius, 77 degrees Fahrenheit, the D. melanogaster lifespan is about 50 days from egg to death. 24. The developmental period for D. melanogaster varies with temperature, as with many ectothermic species. The shortest development time, egg to adult, 7 days, is achieved at 28 degrees Celsius, 82 degrees Fahrenheit. 25-26 development times increase at higher temperatures, 11 days at 30 degrees Celsius or 86 degrees Fahrenheit, due to heat stress. Under ideal conditions, the development time at 25 degrees Celsius, 77 degrees Fahrenheit, is 81/2 days, 25-26-27, at 18 degrees Celsius, 64 degrees Fahrenheit, it takes 19 days, 25-26, and at 12 degrees Celsius, 54 degrees Fahrenheit, it takes over 50 days. 25-26 under crowded conditions, development time increases, 28 while the emerging flies are smaller. 28-29 females lay some 400 eggs, embryos, about 5 at a time, into rotting fruit or other suitable material such as decaying mushrooms and sap fluxes. Drosophila melanogaster is a holometabolous insect, so it undergoes a full metamorphosis. Their life cycle is broken down into 4 stages: embryo, larva, pupa, adult. 30. The eggs, which are about 0.5 mm long, hatch after 12 to 15 hours, at 25 degrees Celsius or 77 degrees Fahrenheit. 25-26 the resulting larvae grow for about 4 days, at 25 degrees Celsius, while molting twice, into second and third instar larvae, at about 24 and 48 hours after hatching. 25-26 during this time, they feed on the microorganisms that decompose the fruit, as well as on the sugar of the fruit itself. The mother puts feces on the egg sacs to establish the same microbial composition in the larva's guts that has worked positively for herself. 31 Then the larvae encapsulate in the puparium and undergo a 4-day-long metamorphosis, at 25 degrees Celsius, after which the adults eclose, emerge. 25-26 Drosophila melanogaster, commonly known as the fruit fly, has been a significant model organism in embryonic development research. Many of its genes that regulate embryonic development and their mechanisms of action have been crucial in understanding the fundamental principles of embryonic development regulation in many multicellular organisms, including humans. Here are some important genes regulating embryonic development in Drosophila melanogaster and their modes of action. Maternal genes. These genes are encoded in the female fruit fly and are present in the early stages of embryo development. They determine the embryo's main features and early development. 4. Synonyms. 2. Show list. Life cycle and reproduction. Example: the gene called bicoid regulates the formation of the embryo's anterior end, and its absence leads to an embryo lacking a head. Zygotic genes. These genes are activated in later stages of embryo development when the fruit fly embryo begins to produce its own genetic products. For example, the hunchback gene regulates the formation of segments in the embryo. Homeotic genes. This gene family regulates segmentation and axial patterning in development. They act as regulatory factors that determine cell fate in embryonic development. For example, the gene called antennapedia regulates the formation of anterior limbs in the embryo. Morphogens. These are molecules that form gradients in embryonic development and regulate cell fate depending on their position in the gradient. For example, the hedgehog morphogen regulates the differentiation of segments and segment identity in the fruit fly embryo. These genes and their modes of action form a complex regulatory network that guides the embryonic development of Drosophila melanogaster. They influence cell differentiation, segment formation, and axial patterning in the embryo, ultimately leading to the development of a fully formed adult fruit fly. 035. Sound of Drosophila heartbeat. Males perform a sequence of 5 behavioral patterns to court females. First, males orient themselves while playing a courtship song by horizontally extending and vibrating their wings. Soon after, the male positions himself at the rear of the female's abdomen in a low posture to tap and lick the female genitalia. Finally, the male curls his abdomen and attempts copulation. Females can reject males by moving away, kicking, and extruding their ovipositor. 32. Copulation lasts around 15 to 20 minutes. 33. During which males transfer a few hundred, very long 1.76 millimeters, sperm cells in seminal fluid to the female. 34. Females store the sperm in a tubular receptacle and in 2 mushroom-shaped spermathecae. Sperm from multiple matings compete for fertilization. A last male precedence is believed to exist. The last male to mate with a female sires about 80% of her offspring. This precedence was found to occur through both displacement and incapacitation. 35. The displacement is attributed to sperm handling by the female fly as multiple matings are conducted and is most significant during the first 1 to 2 days after copulation. Displacement from the seminal receptacle is more significant than displacement from the spermathecae. 35. Incapacitation of first male sperm by second male sperm becomes significant 2 to 7 days after copulation. The seminal fluid of the second male is believed to be responsible for this incapacitation mechanism, without removal of first male sperm, which takes effect before fertilization occurs. 35. The delay in effectiveness of the incapacitation mechanism is believed to be a protective mechanism that prevents a male fly from incapacitating his own sperm should he mate with the same female fly repetitively. Sensory neurons in the uterus of female D. melanogaster respond to a male protein, sex peptide, which is found in semen. 36. This protein makes the female reluctant to copulate for about 10 days after insemination. The signal pathway leading to this change in behavior has been determined. The signal is sent to a brain region that is a homologue of the hypothalamus, and the hypothalamus then controls sexual behavior and desire. 36. Gonadotropic hormones in Drosophila maintain homeostasis and govern reproductive output via a cyclic interrelationship, not unlike the mammalian estrus cycle. 37. Sex peptide perturbs this homeostasis and dramatically shifts the endocrine state of the female by inciting juvenile hormone synthesis in the corpus allutum. 38. D. melanogaster is often used for life extension studies, such as to identify genes purported to increase lifespan when mutated. 39. D. melanogaster is also used in studies of aging. Werner syndrome is a condition in humans characterized by accelerated aging. It is caused by mutations in the gene WRN that encodes a protein with essential roles in repair of DNA damage. Mutations in the D. melanogaster homologue of WRN also cause increased physiologic signs of aging, such as shorter lifespan, higher tumor incidence, muscle degeneration, reduced climbing ability, altered behavior and reduced locomotor activity. 40. Meiosis. Meiotic recombination in D. melanogaster appears to be employed in repairing damage in germ-line DNA, as indicated by the findings that meiotic recombination is induced by the DNA-damaging agents ultraviolet light, 41, and mitomycin C. 42. Females. Mating in captivity. Females become receptive to courting males about 8 to 12 hours after emergence. 43. Specific neuron groups in females have been found to affect copulation behavior and mate choice. One such group in the abdominal nerve cord allows the female fly to pause her body movements to copulate. 36. Activation of these neurons induces the female to cease movement and orient herself towards the male to allow for mounting. If the group is inactivated, the female remains in motion and does not copulate. Various chemical signals such as male pheromones often are able to activate the group. 36. Also, females exhibit mate choice copying. When virgin females are shown other females copulating with a certain type of male, they tend to copulate more with this type of male afterwards than naive females, which have not observed the copulation of others. This behavior is sensitive to environmental conditions, and females copulate less in bad weather conditions. 44. Males. Courtship behavior in male. The male first showed wing extension, stage 1, and later other steps such as abdomen bending, stage 2, then frequent attempted copulation, licking and even ejaculation, stage 3, finally the male fell over and was on its back, stage 4. D. melanogaster males exhibit a strong reproductive learning curve. That is, with sexual experience, these flies tend to modify their future mating behavior in multiple ways. These changes include increased selectivity for courting only intraspecifically, as well as decreased courtship times. Sexually naive D. melanogaster males are known to spend significant time courting interspecifically, such as with D. simulans flies. Naive D. melanogaster will also attempt to court females that are not yet sexually mature, and other males. D. melanogaster males show little to no preference for D. melanogaster males over females of other species or even other male flies. However, after D. simulans or other flies incapable of copulation have rejected the males, advances, D. melanogaster malice are much less likely to spend time courting nonspecifically in the future. This apparent Learned behavior modification seems to be evolutionarily significant, as it allows the males to avoid investing energy into futile sexual encounters. 45. In addition, males with previous sexual experience modify their courtship dance when attempting to mate with new females. The experienced males spend less time courting, so have lower mating latencies, meaning that they are able to reproduce more quickly. This decreased mating latency leads to a greater mating efficiency for experienced males over naive males. 46. This modification also appears to have obvious evolutionary advantages, as increased mating efficiency is extremely important in the eyes of natural selection. Polygamy. Both male and female D. melanogaster flies act polygamously, having multiple sexual partners at the same time. 47. In both males and females, polygamy results in a decrease in evening activity compared to virgin flies, more so in males than females. 47. Evening activity consists of those in which the flies participate other than mating and finding partners, such as finding food. 48. The reproductive success of males and females varies, because a female only needs to mate once to reach maximum fertility. 48. Mating with multiple partners provides no advantage over mating with one partner, so females exhibit no difference in evening activity between polygamous and monogamous individuals. 48. For males, however, mating with multiple partners increases their reproductive success by increasing the genetic diversity of their offspring. 48. This benefit of genetic diversity is an evolutionary advantage because it increases the chance that some of the offspring will have traits that increase their fitness in their environment. The difference in evening activity between polygamous and monogamous male flies can be explained with courtship. For polygamous flies, their reproductive success increases by having offspring with multiple partners, and therefore they spend more time and energy on courting multiple females. 48. On the other hand, monogamous flies only court one female, and expend less energy doing so. 48. While it requires more energy for male flies to court multiple females, the overall reproductive benefits it produces has kept polygamy as the preferred sexual choice. 48. The mechanism that affects courtship behavior in Drosophila is controlled by the oscillator neurons DN1s and LNDs. 49. Oscillation of the DN1 neurons was found to be affected by sociosexual interactions, and is connected to mating-related decrease of evening activity. 49. Model organism in genetics. D. melanogaster remains one of the most studied organisms in biological research, particularly in genetics and developmental biology. It is also employed in studies of environmental mutagenesis. History of use in genetic analysis. Alfred Sturtevant's Drosophila melanogaster genetic linkage map. This was the first successful gene mapping work and provides important evidence for the chromosome theory of inheritance. The map shows the relative positions of allelic characteristics on the second Drosophila chromosome. The distance between the genes, map units, are equal to the percentage of crossing over events that occurs between different alleles. D. melanogaster was among the first organisms used for genetic analysis, and today it is one of the most widely used and genetically best known of all eukaryotic organisms. All organisms use common genetic systems. Therefore, comprehending processes such as transcription and replication in fruit flies helps in understanding these processes in other eukaryotes, including humans. 50. Thomas Hunt Morgan began using fruit flies in experimental studies of heredity at Columbia University in 1910 in a laboratory known as the fly room. The fly room was cramped with 8 desks, each occupied by students and their experiments. They started off experiments using milk bottles to rear the fruit flies and handheld lenses for observing their traits. The lenses were later replaced by microscopes, which enhanced their observations. Morgan and his students eventually elucidated many basic principles of heredity, including sex-linked inheritance, epistasis, multiple alleles, and gene mapping. 50. D. melanogaster had historic